So CRISPR-Cas9 is a DNA manipulation tool that involves directly editing genes within an organism. CRISPR-Cas9 is an enzyme found in bacteria that functions somewhat like an immune system. And so we'll look at this later on in Unit 4. So CRISPR-Cas9 works in bacteria by recognizing viral DNA and cleaving it, therefore protecting the bacterium from the virus. And scientists have actually used this mechanism to edit genomes by making CRISPR-Cas9 cleave DNA at a specific location. Okay, good. I have got a diagram here. I'm just going to spend some time going through this diagram because it's kind of a complex process. Let me just get a drink first. So as I mentioned before, in bacteria, we don't have immune systems. And so when a bacteriophage virus infects the bacteria, what it will do is that it will inject its genetic material into the poor bacteria. That genetic material is going to be transcribed and translated, and it's going to make viral proteins, which will then escape the bacteria and release it into the environment. So the bacteria will get sick, poor bacteria. However, when that bacteriophage infects that same bacteria once again, what you have is the bacteria recognizing that virus, right? And so it actually goes, hey, I've seen you before. And it releases its endonucleases to cut up the invading viral bacteria, so viral DNA or RNA, and it will kind of render that genetic material immobile and useless. And so it can actually cause like hijacking of all the cellular mechanisms of that bacteria and making viral proteins because it's been chopped up. So this is due to the bacteria actually keeping a copy of that viral DNA the first time. Okay, so it gets sick the first time and it goes, okay, I'm going to get sick, that sucks. But I'll actually remember it. So when the viral DNA enters the cell for the first time, a sequence of the DNA, known as a spacer, is actually incorporated into the bacterial genome. So the first time that DNA from that bacterial virus infects the bacteria, it's actually going to be saved in the bacterial genome. The bacteria is going to be like, okay, I'm going to get sick now, that sucks, but I'm going to remember you for next time. And so it makes sure to take a sequence of DNA known as a spacer and save it in its own genome. And it's saved within a particular region called the CRISPR array. Okay? So the bacteria will save the spacer within the CRISPR array for the next time. If the same viral DNA enters the cell at another time, the bacterium will recognize the DNA due to the spacer that matches it. So that virus is going to infect the bacteria again. The bacteria is going to be like, wait a minute, I know you, because it actually has that spacer sequence already incorporated into the CRISPR array. This DNA spacer will then be transcribed. So it's in the DNA with the bacteria. It will then be transcribed, so you've got a complementary sequence. And they'll be transcribed to RNA, something called guide RNA. That guide RNA will actually join something called the Cas9 enzyme. So the Cas9 enzyme is actually an endonuclease, and it's a type of molecular scissor. And so this guide RNA joins the molecular scissor, and it's like a recognition site. It will recognize its particular invading viral genome DNA, and it's bound to the molecular scissors. And so it finds that site of the DNA where it recognizes, so that one that the initial spacer came from the virus, will recognize it because it'll be complementary, so it will bind to it. You can have the Cas9 chopping it up. Eventually, this will cause the corresponding section of viral DNA to be cleaved, effectively destroying the viral DNA and preventing infection. Okay? So I think I might go through that process again. Just like super thirsty. So what happens is that CRISPR-Cas9 is a type of like viral defense system that bacteria have. Bacteriophage will infect the bacteria. It will insert DNA or RNA into it. It will make the bacteria sick, and the bacteria will get sick, which is sad for the bacteria, but it will actually remember that virus. And so it actually saves a copy of the viral DNA into something called the CRISPR array. So we'll save a sequence of viral DNA called spacer and save it within the CRISPR array. Okay? When it gets infected by that bacteriophage, once again in the future, it's actually going to transcribe that spacer sequence to make a complementary strand known as the guide RNA. This guide RNA will be complementary, and so it will actually bind to the bacteriophage's viral genome, the invading genome, and recognize it. And because this guide RNA is actually attached to the Cas9 complex, or the Cas9 enzyme, it will actually chop it up and cleave it. And this will effectively destroy the viral DNA and prevent infection. This is really cool for bacteria. It means they've got kind of a rudimentary immune system. But it's even cooler for us because we've actually kind of stolen this genome technology and kind of exploited it in the lab, sort of. So what happens is that we actually use the system in order to edit genes. And so in bacteria, the guide RNA matches the viral DNA to be cleaved. However, we can actually create a guide RNA for whatever sequence we want. So we can actually get a piece of DNA, examine it, and actually create our own guide RNA which will be complementary to it. So for example, if there is a hereditary disease such as Huntington's disease or sickle cell anemia or something like that, we can actually examine the genome, and particularly the sequence which actually leads to this disease, and we can actually create a guide RNA which is complementary to this particular gene of interest. As such, we can actually create a CRISPR-Cas9 complex, so we'll have that guide RNA, which is complementary to it, and we can attach it to the Cas9 molecular scissors or endonuclease, and it will cut the gene at that particular region. As such, we can actually cut up or cut out that particular sequence which causes the disease. We can actually try and solve quite deadly or detrimental diseases here. So pretty much the options are quite endless. We can cut out a gene and inactivate it, but what we can also do is use other technologies to add a gene, modify a gene, delete more of a gene, and more. So it's actually a really useful process. So yeah, we call this CRISPR-Cas9. So you can see here we've got a double-stranded DNA. So this is the target DNA. We'll have this guide RNA here, which is actually targeting. So the guide RNA here is targeting this particular sequence here, and you have the Cas9 sequence, which actually causes that cleavage process. So it snips up that DNA and causes an incision, which you can then insert another gene into, or delete this region, or modify it, etc. So it's kind of a lot that you can do here. Just taking time. So we discussed endonucleases a few slides ago and how they know not to cleave DNA belonging to the bacterium. Actually, I don't know if I did that or not, but I'll let you know now. Bacterial DNA is methylated, whereas foreign DNA isn't. So if we go back a few slides, here you've got the bacterial genome, and here you've got the invading viral DNA or RNA. And so the endonucleases are going to cut up this invading DNA because we'll actually scan it. And so bacterial DNA is methylated, meaning it's got little CH3 groups, kind of like little flags saying, hey, don't kill me. Whereas the viral DNA or RNA doesn't have this. And so the endonuclease will be like, hey, look, no methylated flags, let's just go off and kill it, okay? So it kind of checks it first. It scans it first with these little methyl groups. And if no methyl groups, then it will cut it up. So as mentioned here, bacterial DNA is methylated, whereas foreign DNA isn't. So endonucleases only cut DNA that they are supposed to. So the CRISPR-Cas9 system has a really similar safeguard, and it's something called the PAM sequence, and this stands for protospacer adjacent motif. This is how the Cas9 endonuclease or restriction enzyme knows to cleave viral DNA, but not the spacer in the CRISPR array. So when you create your CRISPR-Cas9 in the lab, or in a bacteria, you actually have the guide RNA being saved within the actual bacterial genome, right? It's saved within the CRISPR array. What happens though is when you actually get the sequence of interest and save it as a spacer sequence in the CRISPR array, you always make sure to take a segment of DNA which is next to something called the PAM, protospacer adjacent motif. So I'll just repeat that. When the bacteria is kind of saving a spacer in the CRISPR array, it always saves a sequence of DNA right next to something called the PAM, which is the protospacer adjacent motif. So it saves that and it puts it into the bacterial genome in a special region called the CRISPR array. Okay? Yeah, so it saves it in the CRISPR array. It always makes sure to take it from the PAM sequence. Cas9 will only cleave DNA that is followed by a PAM sequence. So when you actually insert it, that spacer sequence into your CRISPR array, you just don't bring the PAM sequence with you. So you snip it out and save it. When you're cutting up with the endonuclease Cas9, it's going to scan through a genome, and if that particular sequence it recognizes is right next to a PAM sequence, then it will cut it up, okay? So it must be followed by a PAM sequence, and it's usually just a short sequence with a few nucleotides, which you can recognize. So the DNA spaces within the CRISPR array are not followed by a PAM sequence, so Cas9 doesn't cleave this DNA, right? It has to be followed by a PAM. Therefore, when we're in the lab and we're actually designing our own guide RNA, scientists first have to make sure that there is a PAM sequence next to the gene that they are targeting. Okay, so you can actually design sequences which will be complementary or bound. So you can actually specifically design guide RNAs, but you need to make sure there's a PAM sequence there. But there are just tons of different PAM sequences, and so you can actually pretty much tailor where you want to cut the DNA. Okay, so you can see here you've got the target DNA, you've got the CRISPR array, or that guide RNA. It will recognize its gene of interest next to a PAM sequence, and so it will cut it with the Cas9, which is that endonuclease enzyme. This cutting it causes a double-stranded break, which is initiated by the Cas9, and you can have insertion of a gene, or correction of a gene, or deletion of a gene. CRISPR-Cas9 is a fairly new discovery. It's been around for a few years now, but it's super useful. It's actually been really involved in many different genome technology processes, which we'll probably discuss. It's a fairly new discovery. Its applications in genome editing are still being studied, and there are lots of ethical laws relating to CRISPR-Cas9 and kind of regulating its use, particularly in humans, but even in genome technologies relating to agricultural needs and farming. And so it is quite contentious at the moment, and there are lots of ethical concerns about it. So some concerns which are being studied are if a gene is inserted, what happens when it's done its job and is no longer required? Just check the time. What if Cas9 cleaves at unwanted locations? Will it work in the long term? For example, people with chronic diseases will constantly need the gene to be working. What if a gene has unknown influence on a certain characteristic or function and is inactivated? What if the mutations caused by CRISPR-Cas9 lead to cancer? The main issue is really potentially off-target effects. Why is that the end of the slideshow? Oh, okay. I'm just going to discuss this kind of casually for 10-15 minutes first, and then we'll just go into kind of discussions about how I study for the exam. So yeah, there's quite a lot of content just about CRISPR-Cas9. I haven't put any articles here, but this is something you'll have to research in your own time. And it's not something you have to research, but I think it's a really good thing to go back and learn more about it. So I would recommend just going into the news, like The Age or my favorite magazine is The Atlantic, and they've got lots of different scientific discovery articles. So what happens is that we can look in these articles for CRISPR specifically and just read up on the ethical discussions. The problems associated with CRISPR. If a gene is inserted, what happens when it's done its job is no longer required? So if you insert a gene, what happens if that gene is no longer needed? Before we were talking about embryonic development, and some genes are only required in embryonic development. And once you finish embryonic development, they're down-regulated or switched off. What if you insert a gene, right, and that gene is not actually needed in the long term? So if you insert this gene and it's kept within this individual in the long term, what happens when you no longer need it? Is it just going to keep producing these proteins you no longer need? Is it going to waste energy and resources producing these proteins? It can actually have adverse effects affecting this individual in the long run. It'd be quite dangerous. What if Cas9 accidentally cleaves at unwanted locations? We know that naturally our body does make mistakes, right? So if we are inserting this CRISPR-Cas9 complex, that guide RNA and the Cas9 endonuclease, into an unwanted location, what happens if it accidentally cuts elsewhere, right? Or what if it accidentally goes to the wrong spot and cuts something which is actually quite vital? What happens then? And so, you may argue that if we can cut up stuff, we can fix it, but it may go unnoticed for quite a long time, right? And it can cause quite a lot of detrimental adverse effects as a result of that. Will it work in the long term? So for example, people with chronic diseases will constantly need the gene to be working. So some people have particular diseases whereby a gene is switched off. What if we use CRISPR to turn it on? What if the gene then gets shut off by another protein or enzyme or something like that? So we really need to make sure that these genes continue to function effectively for the duration. So not just a short period of time, but for kind of a long period of time, particularly long term if someone has a chronic disease. What if a gene has unknown influence on a certain characteristic or function and is inactivated? So you may not know that a gene, you might go, this gene is useless or it's causing problems, so let's turn it off. But what happens if this gene is actually kind of useful? Or what if we don't know everything this gene does? So it's really important to recognize that genes may have many different functions which we don't actually know much about. Like we only fully categorize, or like, yeah, I think it's like categorized the entire human genome a few years ago, like 2 years ago, I think. Because I remember I think it was in second year, and one of my professors made an announcement like, oh my god, we've fully like sequenced the entire genome. So we've sequenced it, but we don't know what every single gene does. There are lots of genes where we think they have no function, or we just think, we've completely categorized its entire functionality. But what if we're wrong? What if there are more functions? And what if switching off this gene can actually lead to adverse effects? Or what if turning on a gene can actually lead to more adverse effects? So there are many off-stream or off-target effects which we perhaps are unaware of right now. Another really big common fear-mongering technique, I guess, is what if the mutations caused by CRISPR-Cas9 can lead to cancer? So what if you accidentally switch on a gene and this causes the creation of a tumor or something? So it's quite dangerous thinking, I guess. I also want to talk a bit more about ethical considerations. So you can use CRISPR-Cas9 for quite a wide myriad of different uses, so most prominently in agriculture and farming. And so you can actually specifically target particular genes. And so the agricultural and farming industry uses CRISPR-Cas9 quite prominently now. So for instance, you can have tomatoes, right? And tomatoes are like fruit in a particular season. And so what you can do is you can actually use CRISPR-Cas9 to modify the genome of a tomato plant and actually make it so that it flowers all throughout the year, as opposed to it's just one like season of fruiting. And so this means you've got tomato plant flowering all year. And so this can be really useful to farmers who actually rely on tomatoes, right? They can actually like have more tomato fruits all year, which might be really useful. However, how does this affect like the wider ecosystem? So tomato plants maybe are supposed to flower only one particular season of the year, because then other plants are able to flower throughout the rest of the year. And so without this, you may actually be disturbing the natural ecosystem of the plants. This might seem like kind of a small scale problem, like, okay, so what? But there might be particular bugs or insects which actually rely on other plants to survive. Or perhaps some bugs rely only on tomatoes, and so as a result, they're actually flourishing. And also lead to an imbalance of these particular bugs which rely on tomatoes, and hence disturb the ecosystems, because then you'll have maybe the pests which rely on these bugs, things such as birds or rabbits or something, maybe. Actually, rabbits don't eat bugs. Birds or something like that, they will then flourish. Or for instance, rabbits may be relying on these tomatoes now, and they'll actually have an increased surplus of rabbits, and as a result, foxes will be well fed, and then it kind of has off-stream effects on the larger ecosystem, you can see. Other things you can use CRISPR-Cas9 for include making things pest resistant. So some plants are particularly susceptible to locusts, or may be really susceptible to disease. And so you can actually make them resistant to disease or pests. And so as a result, they won't die from diseases. But diseases are a method of natural selection. So diseases will wipe out particular plants or individuals who are considered not strong enough to withstand them. As a result, only those who survive have genes which confer like strength and make them immune to like particular diseases, right? And so this allows us to undergo mutations or wipe out members of the population who are considered not strong enough to withstand these things. If we are modifying tomato plants or potato plants or whatever to make them withstand diseases, we're kind of messing with the natural selection of things. And many people don't like this idea of kind of playing God. They think it's unnatural and it's disturbing the natural environment, and it can be disturbing the ecosystems in quite a big way. Some other big concerns relating to CRISPR-Cas9 and other genome modifying technologies include organicness of materials. So there was a big lawsuit a few years ago whereby a farmer who was like an organic farmer, he was saying his fruits were all organic. However, someone next door to him was using genome technologies to kind of alter their plants. And so this actually kind of spread into this organic farmer's seeds. And so as a result, he was sent to produce plants which were no longer organic anymore, because they were actually modified via genome technologies such as CRISPR. And so this is also a problem. It's kind of messing with the ethics of other people. It can spread. And a really big problem relating to this is that related to pesticides. So farmers can actually make their plants or genetically engineer their plants so that they are resistant to pesticides. What this means is that, I can engineer a tomato plant to make it resistant to pesticide. As such, I can spray an entire field with pesticide, and the tomato plants are all going to survive. They're not going to die. So all the weeds are going to die because they don't have the pesticide-resistant genes. But what happens if these pesticide-resistant genes actually spread into the weeds themselves? As a result, you'll actually have weeds which are resistant to pesticides. You kind of have to keep leveling up the amount of pesticide use in order to kill off those weeds. Moreover, pesticide use isn't a good thing anyway. Like, it can be quite detrimental to our health as human beings. So we do want to avoid using them. And so just like spraying an entire field without abandon just to kill off the weeds and using lots of pesticides can cause detrimental problems for us. So we will eat these plants and we might get sick as well. So that's kind of another severe problem. So I've kind of discussed quite a few problems. I might just discuss a few like good things about CRISPR, and then we'll discuss studying for the exam and doing well in bio overall, which I think is probably what a lot of you guys are here for. But yeah, first, benefits of CRISPR. So yeah, we have talked about like quite a lot of negative stuff about CRISPR, a lot about like playing God or pesticides and things. So I might just quickly talk about one more. No, I reckon that's fine. Okay, we'll talk about positives. So a few years ago, I think in parts of Asia, quite a lot of kids or students were going blind. Like their vision wasn't very strong. Like their vision was deteriorating quite quickly and at quite a young age. And so this was due to a deficiency in vitamin A, or vitamin A in their diet, or actually a precursor of vitamin A in their diet. And so scientists and health boards wanted to solve this problem. So what they did is they actually engineered rice. So rice is a staple food in these environments, and they actually genetically engineered rice to actually express a vitamin A precursor gene. And so this meant the rice would actually go like yellow. It would be like a golden color, so it's called golden rice. And so this yellow rice was then distributed across these areas, and because it's a stable food, people would eat it more often, right? Or eat it pretty often. But because it's actually been enriched with this vitamin A precursor, it actually enabled these kids to produce more vitamin A. And as a result, their eyes were starting to deteriorate less. And it actually improved the vision of like entire communities in this area and actually prevented the blindness from occurring at such a young age. So you can see this is like a big success story of CRISPR. It's actually prevented quite a lot of blindness in lots of young people. So you can see it's actually done quite a lot of good. You can also like make vegetables like fruitier, like bigger fruits or more juicy vegetables or tastier, stuff like that. Enrich them in vitamins. So this can also be really good at like preventing sicknesses and disease by enrichment, and it can be quite like a cheap and relatively easy process to do. But you also have to be like really aware of the fact that large companies and corporations may actually have a monopoly on these plants. So a lot of companies will actually genetically engineer plants so that you can't take the seeds and plant them and grow your own plant. And so they pretty much hold the patent or the rights to the entire plant. And they can like hike up the prices of these plants to farmers and make the farmers pay more. And farmers obviously depend on these seeds for their livelihoods. And so you see there's kind of like a large power imbalance being created here. So it's really important to keep that in mind too. Other benefits of CRISPR. I mean, you can look at everything I've just discussed previously on like the other point of view. So, making them more pest resistant, like if we just kind of put aside the fact that you're spraying pesticides, it can be quite useful to create pest resistant plants because then they don't die from locusts and things. So yeah, looking at like both sides and being able to discuss both sides is really important. And I would highly recommend just every few weeks going back and looking up benefits of CRISPR, pros and cons, stuff like that. Cool, okay. That was most of what I had to discuss about CRISPR, I think. Probably a bit more than I needed to discuss.